All right, let's get right into it. We're going to talk about the fundamental rules of life itself, how our cells divide. And by understanding this incredibly complex process, we can see exactly how scientists are learning to hack it, opening up a whole new frontier in the fight against cancer. Just think about that number for a second. Millions of new cells every single minute. Now, this isn't some chaotic free-for-all. It's a super organized, tightly controlled biological dance. But what happens when one of those cells decides to break the rules? Well, that's where our story really begins.
So, to get a handle on the rules, we first have to understand the system that keeps everything in line. You can think of it as the cell's internal clock, but to a scientist, it's known as the cell cycle.
The most important word here is precision. The whole point of the cell cycle is to make a perfect, flawless copy of the cell's genetic blueprint, its DNA, and then make sure that the two new cells each get an identical copy. There is absolutely zero room for error.
You can think of it like a four-act play. Act 1 is the G1 phase, where the cell just grows and gets bigger. Act 2 is the S phase. S stands for synthesis, and this is the main event where all the DNA gets copied. Act 3, G2, is like the final dress rehearsal, one last growth spurt and systems check. And then the finale, M phase, or mitosis, where that one cell dramatically splits and becomes two.
Now, with a process this critical, you know there have to be some safety measures, right? Well, the cell has its own team of quality control inspectors, and they are stationed at these crucial safety checkpoints.
And these aren't just suggestions, they are hard, non-negotiable stop signs. At very specific moments, the entire cycle just pauses. It asks itself questions like, is our DNA damaged? Are we big enough to divide yet? Are all the chromosomes lined up and ready to go? If the answer to any of those is no, everything grinds to a halt until the problem is fixed.
So who's actually running this whole operation? Well, meet the management team. It's basically a duo of proteins. You've got the engines, which are called cyclin-dependent kinases, or CDKs for short. They provide all the raw power. But an engine is pretty useless without someone to turn the key. And that's where their partners, the cyclins, come in. The cyclins are the managers that tell the CDK engines exactly when to turn on and drive the process forward.
And it's not like any old manager can work with any engine. Oh no, the cell is way smarter than that. There's a very specific manager-engine pair for each and every job. For that initial growth phase, you've got cyclin D pairing up with CDK 4 and 6. Then, like a relay race, they pass the baton to cyclin E and CDK 2 to get the DNA copying started. It's this incredible specificity that keeps the whole thing running on a perfectly timed schedule.
So, we've painted this picture of a perfect, well-oiled machine, but now we have to ask the really important question. What happens when this beautiful system of checks and balances completely breaks down?
This quote really just says it all. The loss of control over the cell cycle, it isn't just a side effect of cancer, it's one of its absolute defining features. It's a hallmark of the disease. A cell that can't stop dividing is, by its very definition, on the path to becoming a cancer cell.
Let's talk about one of the most important stop signals that healthy cells follow. It's an elegant, simple rule of cellular society called contact inhibition.
Okay, picture this. You're growing cells in a lab dish. Healthy cells will spread out and divide, but only until they form a perfect, single-cell thick layer. The moment one cell bumps into its neighbor, it gets the message. Okay, space is full. Time to stop growing. They basically respect each other's personal space.
Cancer cells? Yeah, they are the rule breakers. They totally ignore contact inhibition. When they bump into their neighbors, they just keep on dividing, piling up on top of one another, forming these chaotic, disorganized clumps. And that, right there, is how a tumor gets started.
But here's the amazing part. This very breakdown, this broken rule, it actually gives scientists a target. If we know exactly which piece of the machine is busted, maybe we can design a way to jam it.
The logic became crystal clear. If these out-of-control CDK engines are basically flooring the gas pedal on cancer growth, then what if we could design a drug to specifically slam on the brakes for those engines?
Now, the first attempts were a bit like using a sledgehammer. These early drugs, called pan-CDK inhibitors, just shut down a whole bunch of different CDK engines all at once. And while they kind of worked, they caused a lot of collateral damage, side effects. But the real game changer was the next generation, drugs that act more like a surgeon's scalpel. See, in many breast cancers, it's the CDK 4 and 6 engines specifically that are stuck on. So drugs like palbociclib were designed to block only those two. This incredible precision means you get better results with way fewer side effects.
So what does this amazing science, this shift from a sledgehammer to a scalpel, actually mean for real patients? Well, it means something incredibly precious. It means more time.
Let me break down the data for you. In a huge clinical trial for women with advanced breast cancer, adding this one selective drug, palbociclib, to the standard therapy extended the time before the cancer progressed by a median of 10 months. 10 extra months. In the world of cancer treatment, that is a massive, massive victory. This success has completely blown the doors open for a new way of thinking about cancer therapy. It's all about hacking the cell's basic operating system. So where are we today? Well, we now have 3 of these smart drugs officially approved, and the research is getting even smarter. The focus now is on figuring out the best drug combinations and this is really key, finding ways to predict exactly which patients will benefit the most from which drug. This is the very definition of precision medicine, not just treating a disease, but treating your specific version of that disease. Which leaves us with a pretty powerful and hopeful question, doesn't it? As we get better and better at hacking the very engine of cell division, are we getting closer to a future where cancer isn't a death sentence, but instead becomes a manageable, chronic condition? Something you live with, not die from? That's the incredible promise of hacking the cell cycle.